My name is Carissa Ansel, and this is my literature critique of Seroprevalence and Correlates of SARS-CoV-2 Neutralizing Antibodies from a Population-Based Study in Bonn, Germany by Aziz et al. This study was conducted in light of increasing infection rates of SARS-CoV-2. Seroprevalence gives insight to how many people in a community have antibodies against a particular virus. In this case, the virus of interest was SARS-CoV-2. This is a population-based seroprevalence study, and at the time, this type of study was lacking. Participants for this study were gathered from a previous cohort study that was actually paused due to the lockdown of COVID-19. So the study was called the Rhineland study, and participants that were in the Rhineland study were used for this seroprevalence study. There were 4,755 samples that were analyzed, blood-drawn samples, and of those, about 1% tested positive on the first immunoassay test. After that, there were 2 more confirmatory tests to see who had neutralizing antibodies. Of the people who did test positive on the first immunoassay set, 35% had neutralizing antibodies. This was a multi-tiered system for testing people who had neutralizing antibodies against SARS-CoV-2. There were 2 main objectives to this study. First, they wanted to accurately estimate the prevalence of SARS-CoV-2 seropositivity in a region with a relatively low infection rate. The second objective was to investigate the correlates and temporal dynamics of neutralizing antibodies against SARS-CoV-2. The primary outcomes of the study were to evaluate the presence of IgG antibodies against SARS-CoV-2, and then secondly, to examine the length of time that neutralizing antibodies were in the body after first being detected. The main bias of concern with this study is self-referral or volunteer bias. To account for this, what the researchers did was they sampled a group of spontaneous volunteers that were not part of the Rhineland study. And what they found was after formally inviting these individuals, they had less response rate, but the seroprevalence estimates were much higher. So what they found was that people who had knowingly had a COVID-19 infection or had been exposed to COVID-19 were much more prone to come and get tested on the seroprevalence assay. Here you can see a flow chart of the participants in the study. You can see that there were 4,755 analyzed samples, and from those, 36 were ELISA borderline and 46 were ELISA positive. Those that were borderline and positive then underwent more confirmatory testing to see if they had neutralizing antibodies. And circled down in the red, you can see that 16 of the ELISA positives ended up having neutralizing antibodies, and only 1 of the ELISA borderline had the antibodies. Another aspect of the study was to analyze how having neutralizing antibodies affected IgG antibody response. Circled in red are the individuals who did have the neutralizing antibodies. You can see here that they had a significantly higher IgG response because of the neutralizing antibodies. The main conclusions of the study were threefold. First, neutralizing antibodies were detected in 1/3 of those who had the positive immunoassay results, the ELISA results. Secondly, neutralizing antibodies waned quite quickly within just a few months of detection. Thirdly, the main overall conclusion was that without a vaccination, immunity to SARS-CoV-2 is very difficult to gain. One limitation to this study is that it was only done for adults ages 30 and older. Secondly, the researchers did not assess the specific T cell and memory B cell responses for SARS-CoV-2. Therefore, this can't be considered a complete comprehensive understanding of immunity against SARS-CoV-2. In terms of generalizability, this study results can be applied to an adult population age 30 or above. It is more likely that there are false positives since the prevalence was so low in Bonn at the time of this study. Thank you for your time, and I appreciate your comments in the discussion post.